Well, thank you very much for the introduction and the opportunity to join you all today. Sorry I can't be there in person. Um, but happy to join the workshop and talk to you today about adaptive designs. And I'm going to cover some key principles from a regulatory perspective, and then also just touch on a few specific recommendations in areas where I think there could be some improved practices or gaps that could be filled. So here's an outline for my talk. I'll have a brief introduction, cover some key principles, and then go through some recommendations. So just in terms of an introduction, I think, a lot of what I'm going to cover today is out of our FDA guidance on adaptive designs for clinical trials, and I provide a link to that here. So, encourage everyone to take a look at this if they haven't, to get a fuller picture. And then I think, as has been mentioned multiple times during the workshop, there's ongoing work on an ICH guideline to hopefully come up with harmonized international guidance on the design, conduct, analysis, and interpretation of clinical trials within adaptive design. And, similar to the FDA guidance, this guideline will focus on, core principles for regulatory review and for sponsors to consider in designing, conducting, analyzing, interpreting results from adaptive design trials, and really hope that this will publish soon for public consultation. Final slide as part of my introduction. I just want to emphasize that in a given development program, in a given trial setting, the ultimate decisions regarding design elements depend on many factors, both scientific and non-scientific. And this is a core reason why, at least from an FDA perspective, our guidance really focuses on key principles that should be considered, that should be followed, and does not really intend to require or restrict use of specific designs and specific adaptive designs, which is the topic today, in general settings or in specific settings. But that being said, it's really good practice for sponsors to explore the operating characteristics for a variety of design options and to submit that information to discuss considerations related to that evaluation with regulators, particularly when we're talking about planning of confirmatory trials. So I'll touch on some of the core principles in the adaptive design space. And these are the principles that are emphasized in the adaptive design guidance. And I'll go through each one of these in a bit more detail in the coming slides. But the FDA guidance emphasizes 4 principles. The first being control of the chance of erroneous conclusions. The second being the reliability of treatment effect estimates. The third, the adequacy of the pre-specification of the design, and then the fourth, maintaining trial integrity. As I think Frank Bretz noted earlier, there's others that could be included here, including, how well the adaptive design fits within the context of the overall development program. But I'll talk through these 4 that are emphasized in FDA guidance. So in terms of controlling the chance of erroneous conclusions, and this has been discussed, in detail, but I think, at a high level, we want to make sure that both at the trial level and at the development program level, that there's limitation of the probability of making bad decisions, including incorrect conclusions of safety or effectiveness, or incorrect conclusions of lack of safety or effectiveness. And from an effectiveness standpoint, this is typically demonstrated, although not always, but typically demonstrated through a frequentist test of a null hypothesis at a one-sided 2.5% significance level. And it's well known, well established, that adaptive designs can inflate this probability. So this problem can be solved. There is a variety of existing testing methods that can be used where the error control is either supported by analytical theory, or in some cases, there can be sufficient support through comprehensive simulations that the error is controlled. The second principle, is ensuring reliable estimation. So as has been emphasized, many times by previous speakers, testing isn't sufficient on its own. It's also really important to have accurate and precise treatment effect estimates to facilitate a reliable benefit risk evaluation, because our ultimate regulatory decisions are based on benefit risk, and also to ensure appropriate labeling to communicate information to patients and prescribers and enable evidence-based medicine. And adaptations can induce bias in estimates, they can lead to incorrect confidence interval coverage, they can have other consequences on the reliability of estimates. And so the ultimate recommendation is to use methods that adjust for the adaptive design when those methods are available to improve the operating characteristics of the estimates, or at minimum, at least evaluate their performance so that they could be interpreted with caution. And I'm going to come back to some of the kind of thought-provoking questions that were posed earlier about reliable estimation later in the talk. The next principle is just, complete pre-specification. So kind of lists some of the elements of that, but it kind of goes beyond pre-specification in a standard or traditional non-adaptive design. And the additional elements that would be really important to pre-specify and think carefully through at the planning stage in an adaptive design would include the anticipated number and timing of interim analyses, the type of adaptation, the approach to maintain trial integrity, the statistical methods that will be used at the interim and final analyses, and also the anticipated algorithm that governs the adaptation decision. And this latter one is an element that isn't always adequately pre-specified in an adaptive design. So what's the rationale for this degree of planning, this degree of pre-specification? Well, for some types of adaptations, it's important because it just facilitates the use of appropriate inferential methods. But even if this degree of pre-specification isn't essential for, say, type 1 error control, it's still important because it increases confidence that adaptations were not based on accumulating knowledge in an unplanned way. It facilitates the understanding of the trial operating characteristics. It reduces the desire for sponsor access to comparative interim data. It ensures the data monitoring committee, if it's involved in the design, can focus on patient safety and trial integrity. So there are lots of reasons for this degree of pre-specification in an adaptive design. And then the fourth principle emphasized in FDA guidance is just the importance of maintaining trial integrity. And this isn't restricted to adaptive designs. It's recommended in all trials, whether adaptive or not, that access to comparative interim results is limited to individuals who are independent of the personnel conducting or managing the trial. And the primary motivation for this is that knowledge of interim results can affect aspects of trial conduct, things like patient accrual, adherence, retention, endpoint assessment, in ways that are difficult to predict and really impossible to adjust for. Also, in adaptive designs in particular, limitation of access of such results can also provide greater confidence that potential unplanned design modifications are not motivated by accumulating data. So, for example, if there's a well-motivated protocol amendment based on emerging external information, when a sponsor proposes that, they're in essence protected by not having access to unblinded data from the ongoing trial when they propose such a well-motivated external information-based change to the trial. There are some additional challenges to kind of limiting access and maintaining trial integrity in an adaptive design. And so how can this be achieved? Well, it's generally done through the use of an independent group like a data monitoring committee, where access to comparative interim results is limited to that independent DMC and then the independent supporting group that is actually running the analyses, providing the interim reports for the monitoring committee, along with the use of safeguards to ensure that those people who prepare the interim analysis results are physically and logistically separated from the personnel tasked with managing and conducting the trial, the use of things like confidentiality agreements, firewalls, data access plans, whether these are just specified in the protocol or the DMC charter or in a separate document, at least having careful plans about who will have access to confidential data, when that access will occur, and what types of data and results will be involved. So thinking through all these things carefully to maintain trial integrity, especially in adaptive designs where the adaptations are based on unblinded results, comparative interim results, where treatment groups are compared. So that's kind of my just overall snapshot of, core principles emphasized in FDA guidance. Again, those are elaborated in more detail in the actual guidance document. And I'm going to shift to just a few recommendations for areas where I see some potential for improved practices or other changes in the adaptive design space. So the first recommendation is in the area of estimation. So the recommendation is to use treatment effect estimators that adjust for the adaptive design to improve the reliability of those estimates. I think, as Frank Bretz noted earlier, this is not common. It's pretty standard to, conduct a group sequential or an adaptive design, have careful planning of a testing approach that controls type 1 error, and then just to ignore the estimation problem and report conventional point estimates and confidence intervals in the clinical study report, in the labeling, and in major journal publications. And this is problematic because the typical estimators, for example, maximum likelihood estimator like the sample mean, are biased in the case of adaptive designs. There's lots of publications on this. I cite one here that showed that in simple sample size adaptation designs, you can have the sample mean having considerable bias and up to 40% higher mean squared error than adjusted estimates in that setting. And I think this paper by Robertson and colleagues was cited earlier. It's a really nice summary of the many existing methods that are available. And the general recommendation I would have is that one of these should be pre-specified and used, or at minimum, the extent of the bias should be evaluated. Frank, I think, posed some really challenging and interesting questions earlier on what criteria should be used to evaluate estimates that I don't have answers to. But I would say that I guess, maybe we can address at least the low-hanging fruit, which is in very simple adaptive designs, like a group sequential design or a sample size adaptation where you have a single primary estimand. In those cases, I think we could use one of these existing methods, and they would have considerably better performance with respect to operating characteristics that I think we would all agree is important, including bias, variability, and mean squared error. And as a starting point, I think using one of those methods in those simple cases would get us in a better place. And we could also, at the same time, have continued conversations about some of those more complicated questions and more complicated designs. Second recommendation is in the simulation space. So when simulations are critical to understand trial operating characteristics, the simulations that are conducted need to be rigorous. And it's not that uncommon that we'll see simulation report that kind of talks through a simulation that was conducted that either has a very limited coverage of the parameter space or has a wholly inadequate number of simulated trials to have sufficient precision, or has some other major deficiency. And so it's really important that there's good simulation practices that comprehensively cover the plausible space of treatment effect and nuisance parameters. So considering nuisance parameters such as the variance, the enrollment rates, the dropout rates. There are going to be computational and other challenges. These can, to a certain extent, be addressed through, leveraging clinical considerations about what aspects of what parts of the parameter space are just clinically impossible, using a grid of assumptions, leveraging monotonicity arguments. But nevertheless, there should be a convincing justification that the plausible space of the parameters has been covered by the simulations. It's also important to compare to alternative designs with respect to a comprehensive set of operating characteristics so that we can have a good understanding of the advantages and the limitations. This can be really important, I think, as Andrew Thompson just mentioned, when there's a lot of complexities to understand what the rationale is and what the advantages are. There should be a sufficient number of iterations of repeated clinical trials simulated to have sufficiently precise estimates of the operating characteristics, and having a comprehensive report of the design and the results of the simulations is also important. The third recommendation is to consider more frequent use of group sequential designs in rare diseases. I think this is pretty important. It's really critical in rare diseases to minimize the chance of the trial producing inconclusive results. And when I'm thinking about inconclusive results, I'm thinking about a trial that when you complete it, for example, has a confidence interval that includes both 0 and very meaningful effects. Now, that's a trial that was almost a waste of time because those results are inconclusive, and it's particularly in a rare disease space where there's few patients, considering some of the importance of getting reliable, conclusive results out of such a trial is, I can't underscore that enough. In the rare disease space, there's often limited early phase data, and there's considerable uncertainty in the potential effect size. And so the sample sizes in a rare disease space are often selected to provide adequate power under overly optimistic and sometimes completely arbitrary effect sizes that are determined based entirely on what sample sizes are considered feasible or achievable by the sponsor. So I think this is a really ripe space for the use of group sequential designs and/or designs that incorporate sample size adaptation that allow the possibility of the trial proceeding to a larger maximal sample size to ensure that the trial not only has power under the initially hoped for effect size, but also would have at least some degree of power under smaller effect sizes that are probably plausible and would still be quite meaningful to patients and to public health, while including stopping rules to ensure that the trial will stop under smaller sample sizes if there's large effects, if there's persuasive evidence, and that it will continue on if the interim results are promising but not yet conclusive. So I think this is a space where, it would be really helpful to better utilize some of the simple adaptive designs, or group sequential designs in particular, to provide more conclusive results. My last recommendation is not necessarily restrictive to adaptive designs, but I thought I would throw it in there is, I think there's a lot of opportunity in the master protocol space when those are applicable. Master protocols can meaningfully improve efficiency through shared design and operational aspects. I also think there's a lot of value in umbrella and platform trials and their ability to provide direct comparisons between products to inform treatment decisions in the real world. And also, when there are careful design and analysis approaches utilized in a master protocol, they can provide reliable evidence. And I would point everyone to a recent FDA draft guidance on master protocols that talks through some of those design and analysis considerations for ensuring that they can produce reliable data on safety and effectiveness. I do want to note that a master protocol does not necessarily involve adaptive design. So you can have a master protocol without, a non-adaptive design in a master protocol, but they often at least include interim analyses with the potential for early stopping. So I thought they fit within scope enough to include this slide. I did find the talk earlier today, by Jonathan Kimmelman kind of thought-provoking and reflected on this slide a little bit. I'm not very precise here in my definition of efficiency, but I think with a precise definition of efficiency, I do believe that master protocols would hold up to this conclusion here. And I guess I would also emphasize that not only do some of the design and analysis approaches emphasized in our FDA guidance improve the reliability of the evidence that is generated, I think they also ensure that the results are interpretable. So I think some of the scrutability discussion earlier, I think also applies here and is additional motivation for, while master protocols have a lot of value, they don't necessarily need to be super complex. I think there can be very large, simple master protocols, recovery comes to mind, that can produce very actionable, interpretable evidence. So when I talk about master protocols, I don't necessarily mean that they need to have super complex adaptive designs or other complexities. They can be simplified and still provide efficiencies and reliable evidence. So I think that concludes what I wanted to share today. I'll stop there, and thanks again for the opportunity to participate. Happy to answer any questions.
So you probably can't hear, but there was a lot of applause in the room, just so you. And so we have time for questions, and you have to use the mic though in order for the speaker to hear you.
Thank you. Can I pick up on your recommendation 3 to use more group sequential designs in rare diseases? I mean, whenever I'm advising people, I always tell them you need to be able to recruit the maximum number of patients that the design tells you. And so if I struggle to get 100 people, a group sequential design will tell me you could need 110 people. So I would even get less often a useful answer in that case. So could you maybe sort of elaborate on your thinking there?
Yeah, so I guess my thinking is that the 100 number is not usually a red line. I think it's more, well, you know, we would have a hard time getting 40. We might be able to get 50. Might be possible to get 60 if we spend a couple years recruiting. It might be possible to get 80 if we spend 4 or 5 years recruiting. I think it's usually more along those lines. So yes, if you want to, you know, if you want to achieve a particular power to detect a particular effect size, you compare a fixed sample design to a group sequential design. The group sequential design would need to proceed to a slightly larger maximal sample size. But I think where either a group sequential design or an adaptive sample size estimation or both would provide value here, it would essentially, there's kind of I guess, a push and a pull. Yes, you have to go to a larger maximal sample size in order to be able to provide power to detect slightly smaller but still meaningful effect sizes. What the benefit of that is that if the effect size is as large as what was put into the protocol, what is often put into the protocol, you won't need to go to that larger sample size. You would stop early. You would have a high probability of stopping early. So in essence, there's some safeguarding. I would also encourage futility stopping rules so that you're not going to that large stopping rules if the results aren't sufficiently promising. But I think there's some safeguards for the sponsor in the sense that you are only proceeding to those larger sample sizes, which are going to take more time to enroll, more costs, are going to be more challenging. If the interim results are promising, but not conclusive. But yes, you do need to be able to enroll those sample sizes. I guess I'm thinking of spaces where while getting to those larger maximal sample sizes is difficult, it is possible. So I'm not talking about a disease where there's 10 patients in the world and you really can only get 10 or fewer, but maybe where there's a few thousand and 100 is difficult, but it's achievable and the sponsor comes in with 50. Those are the kinds of settings where I'm commenting on. I don't know if that helps, but it's a good question.